we decrease it, aggregation will be slower. And this indicates that if we increase the propensity of a sequence to form the beta sheet structure that is stable, stabilizes an amyloid, then aggregation will be faster. So again, something intuitive, because amyloid fibrils are stabilized by beta sheet structure. And the third correlation was with charge. This is the net charge of the protein. If we decrease the charge, aggregation will be faster. If we increase the charge, aggregation will be slower. And you know very well that there are, if we increase the net charge of a protein, there will be repulsions. If we decrease the net charge, if we bring the protein closer to neutrality, aggregation will be slower. So the Coulombic force will be lower, and that's why aggregation is promoted when you close the net charge of the protein to neutrality. So 3 correlations, all of them were intuitive, nothing really surprising here, but what we did, based on these 3 correlations, we could edit quantitatively an empirical formula to determine the change of aggregation rate upon mutation as a function of all these 3 parameters, as a function of the change of hydrophobicity, this parameter, and charge. And this is the formula that we edited at that time. You can see the calculated change of aggregation rate upon mutation as a function of the change of hydrophobicity times a number, which is just the slope of this correlation, plus the change of beta sheet structure times a number, which is this slope, minus delta charge times a number. This formula was empirical, okay? It was phenomenological. It was not based on first principles. It just came from the 3 correlations that we got. And this was edited only on the mutations of one protein, human muscle acylphosphatase. But we wanted to see whether it was applicable to other systems. And so we looked at the data in the literature. We didn't do any new experiments here. We just compared the calculated value using our formula with those values that we could get from the literature. And we could see that mutations of amylin, mutation of prion peptides, mutation of alpha-synuclein, all of them could be reproduced, could be explained by the formula, because the calculated value was similar in many cases to the theoretical value. Some cases, like this mutation, the agreement was not good. You can see a considerable difference, but in most cases, there is a good correlation. And you can better appreciate the correlation, the comparison between calculated value and experimental value by looking at this plot, where we have a slope close to 1, indicating that these 3 factors have the right weight in determining the change of aggregation rate following a mutation. So hydrophobicity, beta sheet propensity, and charge of a sequence are important for amyloid fibril formation. And on these grounds, we edited another algorithm to predict the regions of the sequence promoting aggregation and forming the cross-beta core in the amyloid fibrils, which is called aggregator. Actually, we put it online for some time, but then it was sold to, the patent was sold to a company, and they silenced the online, so it's no longer available online. You have to ask the authorization to run it. But this algorithm actually uses the same principles of hydrophobicity, beta sheet propensity, and charge to edit the aggregation propensity along the sequence. This is the amyloid beta peptide, only 40 residues from the N terminus to the C terminus. And you can see that we have 2 peaks. These 2 regions of the sequence, 1521 and 3142, are predicted to have a high aggregation propensity, just based on this aggregator algorithm. And we compared the data with the experimental ones that we had at that time. Actually, it was 2004, 2005, so there were no structures available with solid state NMR or cryo-EM, because because these were developed later. So just a few EPR, some qualitative solid-state NMR, hydrogen deuterium exchange, but you can see that we have an overlap between the regions theoretically predicted and those that were experimental. So on the basis of that, we edited this algorithm. To be honest, there was another group in Heidelberg, the Luis Serrano's group, that edited another similar algorithm based on the same principles, but they did it independently of us. So we paralleled the aggregator, and this other algorithm was the first one, more or less edited independently of each other. But following these two algorithms, there were about 35 algorithms to predict the aggregation propensity. And in my experience, all of them work properly. I mean, all of them are reliable. Some are more reliable than others, but I would say that all of them have a good prediction power. Okay, but it soon emerged another problem. We have polymorphism. We discussed about it. I mean, it's not only a single amyloid structure. We have polymorphism. So what are we really predicting here? Because we have different structures, different amyloid structures. Indeed, this is a structure that was deposited at the PDB. We have seen this structure already. It's a structure of an amyloid founded by A-beta. But this is only one of many different structures. All these structures are real, because all of them were either extracted from the patients or analyzed in vitro using A-beta. So all of them are real. All of them have been solved using solid-state NMR or cryo-EM, and all of them have been deposited in the PDB, the protein data bank. So you can see different structures. Some of them are similar, but some of them are very different. So the question is, okay, we have an algorithm to predict the regions of the sequence that form the beta sheet structure in the amyloid fiber. So we have a very good instrument. But because of this polymorphism, what are we really predicting? Are we predicting an average of this structure, only one structure, the most stable structure? It was challenging. So what we did very recently, actually we published that in 2025 with these two guys that you can see in the picture. We tried to analyze all the 30 experimental structures of A-beta fibers, because 30 are those deposited in the PDB. So all of them were analyzed together in a statistical manner. And we edited this parameter, F-beta N, which is just the fraction of PDB structures having residue number N in a beta sheet conformation. So by comparing all the structures, for example, residue number 32, if residue number 32 is never in a beta strand in the 30 structures, F beta 32 will be 0%, because that residue is never in a beta sheet structure in any of the 30 beta sheet structures. If residue 32 is in a beta strand in 12 out of 30 structures, 12 divided by 30 is 40%. So F beta N in that case is 40%. In other words, F beta N represents the preference of residue N to adopt a beta sheet conformation in the amyloid universe of the A-beta peptide. It is a measure of the propensity of that residue to adopt a beta sheet structure, considering all 30 amyloid structures deposited in the protein data bank or amyloid atlas. And so we can produce again a profile, but this is not theoretical, this is experimental. It's the F beta N profile for A beta, which is F beta N on the y-axis, versus residue number. And you can see, for example, that residues in these regions of the sequence is 60%, indicating then that these residues adopt a beta sheet structure in 60% of the 30 amyloid structures. Here we have 10%, meaning that only 10% of the 30 amyloid structures, only 3 out of 30, 10% is 3 out of 30, have those residues in a beta sheet structure. So, in other words, you can have a profile of the conformational preference to adopt a beta sheet structure. And if we compare this F beta N profile with the theoretical one using Ziggregator, again you can see a similar profile. So the Ziggregator algorithm predicts the regions of the sequence adopting beta sheet structure. And this is still correct if we consider all the amyloid structures. So all the distribution of the amyloid structures that A beta can form. So maybe it cannot predict a single structure because we have polymorphism, but that can predict the distribution of structures that we have. And it works also for alpha-synuclein. For alpha-synuclein, even 89 PDB structures. And you can see the comparison between the F beta N profile, which is an experimental measurement, with Ziggregator. And you can see a colocalization of the peaks in most cases. And also for tau. In tau, we could see just a discrepancy here because there are 2 peaks theoretically, only 1 peak experimentally. So maybe experimentally we have only 1 region and we have just 2 peaks. But you can see that there is, on average, a very good agreement. So in other words, these algorithms can still predict the regions adopting a beta sheet structure, provided you consider the whole distribution of structures, not just one, the whole distribution of structures. Okay? And of course, Ziggregator was our favorite algorithm because we developed ourselves, but we also checked other algorithms like AgreScan, Tango, Vals, Pasta. Pasta actually was edited in Italy by a group of physicists in Padua, Antonio Trovato, Flavio Seno, probably you know this, some of you know these people, and that works also very well. So in my experience, all these algorithms work very well because they are based on similar principles, so it's not surprising that they can work very well. So the conclusion number 1, amyloid fibrils are characterized by structural polymorphism. Despite amyloid polymorphism, individual residues have conformational preferences, and residues with high preference for amyloid populate beta strands more often than others in the distribution of experimentally determined amyloid structures. And so the peaks of the experimental F beta N profile, and I call it experimental because it comes from the structures, match those identified with predictive algorithms of aggregation propensity, edited well before the position of structure in the PDB amyloid atlas. Okay, and then I will go back to some introduction because so far we have talked about structures, amyloid structure, but what about the mechanism? How does a protein convert from its soluble monomeric state into amyloid? So the pathway that many proteins follow is this one. We start from a soluble state. First, we have to form a nucleus. A nucleus that is formed by 2, 3, 4, 5 molecules. In this image we have 3, but we can have more. Sometimes even 1 molecule can be a nucleus. And we have a step called primary nucleation. So the nucleus is an unstable state, but is competent for fibril elongation. As we have a nucleus, then we have elongation. So monomer add on the end of a growing fibril. We can also have dissociation, of course, which is the opposite process. And we also have fragmentation. A fibril, when it's formed, can fragment, and this is a bad sign, because when a fibril ends, we start from one fibril that have two growing ends, one here, into two fibrils, with four growing ends, one here, another one, and another one. So fibril fragmentation is something that can amplify fibril formation. We also have secondary nucleation. Actually, this was developed by Thomas Knowles, who was a physicist, again, at the University of Cambridge. Thomas Knowles found that we could, in a collaboration with Sarah Links, to be honest, he found that on the surface of a fibril, monomers can come together and they can form a nucleus. A nucleus catalyzed by the surface. A nucleus formed on the surface of the fibril. So the fibril surface can act as a catalyzer for nucleus formation. And these fragmentation and secondary nucleation are called secondary pathways because they can amplify fibril formation and they form independently of primary nucleation. Okay, this is a more complex picture showing again native amyloidogenic early aggregates, nuclei, but following three different pathways that have been accepted so far: nucleated polymerization, nucleated conformational conversion, native-like aggregation. I don't want to get into the details, but just say that you can have nucleus formation from a monomer in the first pathway here. You can have anamorphous aggregates that convert into a nucleus in this blue pathway, and native-like aggregation, where you have native-like aggregates that convert into a nucleus. So the process is very complex. Then you can have fibril fragmentation, secondary nucleation, and so on. And this is another study that we did again a long time ago, showing that with another protein that was called IPFN, one can convert this protein into oligomers and then fibrils. And then we wanted to see where the toxicity comes, because in the process of amyloid fibril formation, you have the final end state, which is the amyloid fibril, but you also have the precursor states, like the oligomers, the intermediate states, like the oligomers. So, what is the most toxic? In this experiment, aggregation of hyperfen was induced in vitro, and at different time points, aliquots were added to the extracellular medium of culture cells. So in other words, we used amyloid fibril formation in vitro, but then we tested the oligomers or the fibrils or even the native state with culture cells to see which species was toxic. This was an experiment that we did in a collaboration with Massimo Stefani, who has now retired, a very valid collaborator, and we could see that the aggregates are toxic. This is an assay of toxicity, the MTT reduction assay. Basically, I can explain a few words. Basically, if the cells are healthy, they will reduce MTT to 100%. If they are suffering, they will reduce MTT to a lower percentage. And you can see that the aggregated protein is more toxic than the native protein, which is not surprising. But at this time, when we have only oligomers in the sample, we have the highest toxicity. As the fibrils grow, you can see that toxicity decreases, and at this time, when we have only fibrils in the sample, we have a toxicity similar to that of the native state. You can see that the oligomers and the fibrils were imaged using transmission electron microscopy, like here, and also AFM, and actually these pictures come from Annalisa's AFM, because I have a long-standing collaboration with Annalisa, so even at that time we could detect these And then aggregation of hyperfen was induced in vitro, and at different time points the aggregates were injected into rats' brains. And this is the toxicity of the buffer, no toxicity, the toxicity of the native state, no toxicity. The fibrils have some toxicity, but the highest toxicity comes from the intermediates, oligomers, with a clear dose dependence. Okay, and again, Annalisa came to help us in this other study, where we tried to characterize structurally the oligomers using these two different conditions, condition A on the left and condition B on the right. We found that both conditions that are very different in terms of pH, organic solvent composition and so on, but we found that both conditions could convert hyperfen, the protein hyperfen, into oligomers. And the oligomers were similar in terms of shape, at least from the images that Annalisa provided. We can see a similar shape, a similar height, so a similar size and shape of the oligomers. But surprisingly, only one species, only one oligomer was toxic. This is again the MTT reduction test, untreated cells, no toxicity. IAPP oligomers, toxicity, this is a positive control. A-beta oligomers, toxicity. Hyperfen oligomers under condition A, we have toxicity, similar to our positive controls. Hyperfen oligomers B, we don't have toxicity. So it looks like that we have very similar oligomers because they are indistinguishable in terms of morphology, but only one is toxic. And so we thought maybe, I mean, they look identical, they look indistinguishable in terms of morphology, but maybe there are fine structural differences that explain why only one oligomer is toxic, whereas the other is benign. And so we labeled all these oligomers with specific probes for both the toxic type A in red and the non-toxic type B. In this case, we labeled the residues with prion, which is a probe of spatial proximity, and we found, that in the non-toxic type B oligomers, there are 3 peaks, one here, one here, one here, indicating that these 3 regions of the sequence are structured in the oligomers, and they are in close touch with the same region in an adjacent molecule. And there are differences because you can see that in the red, the peaks are still present, but they have a lower intensity. And this indicates that we have an overlap with the neuropathy profile because all these regions here overlap with the hydropathy profile. So regions forming the structure of the oligomers colocalized with the hydrophobic peaks. And so to explain that, in the non-toxic type B oligomers, the 3 main hydrophobic regions are structured and buried. In the toxic oligomers, the same regions are less structured and solvent exposed. Another determinant was found with size, because in this case we used chaperones to treat oligomers. This is again a picture from Annalisa of the oligomers in the absence of chaperones. When you add chaperones, the oligomers cluster together. If you add alpha B crystalline, which is another chaperone, they cluster together. Actually, if you have a high magnification, you have the impression that you have a clustering of the oligomers. And this indicates that chaperones can cluster these oligomers, but they can also decrease the toxicity, because in this form, when the oligomers are clustered, or in these forms, when the oligomers are clustered, they are no longer toxic. Only in this form, when they are individual, when they are separated, they are toxic. So it looks like that when the oligomers cluster together, the toxicity disappears. So we can conclude, conclusion number 2, the structural determinants of protein oligomer toxicity. We could identify two reasons, two structural reasons why toxic oligomers are toxic and others are not that toxic. The exposure of hydrophobic residues is responsible for toxicity, and the small size is responsible for toxicity. A cartoon with toxic oligomers with hydrophobic groups exposed, and in the non-toxic groups, these of course are still present because the protein is the same, but the toxic groups are buried. So they cannot make mis-interactions with the surrounding system. And the small size, if we have toxic oligomers, if we have small oligomers, they are toxic, they come together, they are no longer toxic, but even if they convert into fibrils, they are no longer toxic. So it looks like that the fibrils, the end point is toxic, but the intermediate oligomers are the main culprits, the main species responsible for toxicity. Okay, we can skip that because it was another argument, and this actually summarizes the difference between toxic type A oligomers and non-toxic type B oligomers. They are similar in terms of I, determined with AFM. They both contain beta sheet structure, but they are more structured. The toxic species are more structured than the non-toxic species. They have a structure N-terminus, a flexible N-terminus, solvent-exposed hydrophobic residues for the toxic species, buried hydrophobic residues for the non-toxic species. Okay, and in this case, of course, in terms of size, they were similar, but if you enlarge the size, you have a decrease of toxicity, because that is another determinant. It's very nice to have a seminar with all these things in front. Each time I pass through here, I dream about what is coming next, and it's very nice. Okay, this is another study that we did in a collaboration with Alfonso De Simoni. Actually, we played very little role in this study, so all the credit goes to Alfonso and Giuliana Fusco. We only did some cell biology experiments, but in this case, we compared the non-toxic alpha-synuclein oligomers, and Alfonso found that they can attach to the membrane, but only touch the membrane. On the other hand, the toxic alpha-synuclein oligomers, they are more structured. They anchor using the alpha N-terminus, which adopts an alpha helical structure, and this allows the beta sheet structure core to dig into the B layer, and that's why they cause toxicity. Okay, and then we talked with Annalisa before coming here, and she said maybe you can also have a few slides on phase separation, because the problem of phase separation is becoming very fashionable, because it's increasingly recognized that proteins can form solid inclusions, that's right, but also liquid assemblies, liquid droplets. And we call it liquid-liquid phase separation because the droplets are liquid and the surrounding solution, of course, is liquid. So there are two phases, both are liquids, but they are separated. This is the conventional protein aggregation. So there is nothing new in this slide. You can see the conventional protein aggregation from the monomer to the disorder aggregates, beta sheet aggregates into a fibril. But recently, people are considering another process that can be very important, at least for some proteins. We have evidence that tau alpha-synuclein and TDP43 can form this species. LLPS, liquid-liquid phase separation-driven protein aggregation. In this process, the monomer assembles to form liquid droplets. And in the droplets, the molecules have the characteristics of a liquid state. So they can diffuse exactly like the water molecules diffuse in a glass of water. Okay? So we have liquid droplets where the molecules are disordered. We have low affinity interaction, multivalency, because the molecules can diffuse into the droplets, so they can change partners in the interactions exactly as water molecules do in a liquid state. Liquid droplets can be formed in vitro using these proteins, but also in the cells there are membraneless organelles that are known to be liquid, like stress granules, paraspeckles. I don't know if you have any of these names, but in the literature they are very well described. So liquid-liquid phase separation is something that you can have in vitro with a purified system, but also in the cells. And the liquid droplets concentrate the molecules. So at some point there can be a nucleus, a structured nucleus with beta sheet structure that form within this droplet, where we have a high concentration of molecules. And this nucleus can go further to form a fiber. So this is a model that people are considering. There is some evidence that TDP43, FUS, and other proteins can actually form fibrils in this way, particularly in the cells. And so we started to study phase separation, which is, again, a very important process in biology, but the basis is only physical, because it's a physical process. So in this case, we used this protein, TDP43, which is associated with amyotrophic lateral sclerosis. And using only this domain, the C-terminal domain, which is easier to handle, we tried to make droplets. We could observe the droplets with confocal microscopy, because by labeling the protein with fluorophore, you can visualize with confocal microscopy. What you see here is a confocal microscopy image, where you can see all these droplets, which are round-shaped. All the green droplets that you can see, they are various sizes, but they are all round, and they are phase separated TDP43. Of course, the question is whether they are liquid, because if you see assemblies of this type, they might well be solid. So the round shape is an evidence because of the surface tension. They are round, but another evidence that they are liquid, we used fluorescence recovery in FRAP. I don't know if you are familiar with FRAP. FRAP is fluorescence recovery after photobleaching. What you do in this case, you start from a droplet, you focus on a droplet, and you provide a strong laser to bleach the fluorescence of the fluorophore. So you can see this dark area in the middle. This dark hole in the middle is not absence of protein. It's just the fluorescence of the probe that is bleached. But of course, the black hole contains proteins. And then, after the laser, you measure the time, 1.5 seconds, 3.0 seconds, 4.5 seconds. You can see a recovery of fluorescence, and after 15 seconds, you don't see any bleach. And this is because you have a liquid state where you have internal diffusion, and so the bleached molecule in the middle are replaced by the surrounding molecules that are not bleached, and so you can have a recovery. You can also try to make a plot, you make that statistically. This is the pre-bleach fluorescence, this is the post-bleach fluorescence normalized to 0 and 1, and you can have a recovery where you can determine the recovery in terms of the rate constant for recovery, 0.43 seconds. And the recovery is nearly complete, 99.6%. And the third piece of evidence is coalescence. coalescence, because if the droplets are liquid, they should be able to fuse, to coalesce. And this is a movie where you can see two droplets. You can come back. You can see two droplets that come together and they fuse into a larger ones. Maybe the final one is even too big, but this is because with confocal microscopy, you never know where the plant is, so a droplet can be small, but it's bigger than it appears because of the plant where you observe it. And so in the end, we obtain evidence of a round shape, number one, fluorescence recovery in FRAP, number two, and coalescence. And these three are considered by the international community as the three gold standards for liquid-liquid phase separation. So if you have a protein in vitro and you have these three pieces of evidence, this is enough to claim that you have a liquid state. Purists of liquid-liquid phase separation say that if you have only two pieces of evidence and you miss the third one, it's not a real liquid-liquid phase separation. So you need to have all three. And the method that we are trying to devise, we are very excited about it. So we are showing data that are unpublished, and actually I'm seeking the help from the physical community because we need physicists to interpret this data. So if you also are interested in that, we can talk about it after the seminar. So we tried to do three-dimensional FRAP. We did the three-dimensional FRAP with the super-resolution STED microscopy, which is a confocal microscopy, but it's a super-resolution, so you can get the details even in the 3D. And we studied a droplet that is deposited on a substrate. Like you can see a water drop on the right that is deposited on a leaf. It's not a sphere, but it's an hemisphere. And this is because of the surface tension and the gravity and so on. So actually, when we have a droplet reconstructed with this 3D FRAP, we can have something like an hemisphere. You can see in this movie, actually, you can see in this movie how the fluorescence is distributed in the droplet. So it looks like an hemisphere. After the bleaching, because of this three-dimensional FRAP where we can reconstruct the three-dimensional by looking at different planes, we can see the volume and the shape of the area that is the area that is bleached. And you can see again in the movie that the area that is bleached is not homogeneous, it's very large on top, is very large on bottom, and this is how confocal microscopy works. But with this 3D FRAP, we could quantify the volume of the droplet, the volume of the bleached area, the volume and the unbleached area. And we can do some position-dependent 3D FRAP by moving the region of the bleached area in the center, on the side, at the margin, half of the sphere, even the entire sphere, and we can have we can measure recovery of the FRAP. And so by changing all these parameters, we would like to determine the diffusion coefficient of the molecules to see whether the molecules come from the solvent outside and all these things. And but of course, to do that, we need physicists. Okay, and very quickly, because they told me that I have only 4 minutes, 5, it was 5 1 minute ago, so 4 minutes. Eventually, we did some drug discovery, squalene and trodescobine, two natural molecules isolated from the sharks. So we determined I mean, these are from my colleague in the United States, Michael Zaslow. He studied well before us, in 1993 and 2000, they isolated these two molecules from the gut of the shark, and they, starting from 2017, we started to study them to protect against the neurodegenerative diseases. So, I can skip this slide, okay, and again, something with the help of physicists, we compare squalane and trodescomine. We found that trodescomine insert into the B layer. With QCM and molecular dynamics, we found the portion of the molecule that is inserted, even the angle through which it is inserted. We have also a molecular dynamics movie that shows how trodescomine is inserted. Is actually these orange things, and here you can see the molecule that is inserted with the spermine moiety coming outside. This is molecular dynamics, but the data were confirmed by Quartz QCM. And Annalisa and Riccardo measured the indentation force of membranes without trodescomine and membrane with trodescomine. They found a lower indentation force and a higher indentation force in the presence of trodescomine. So a higher indentation force is a consequence of the addition of trodescomine. A lower negative charge is another consequence, and a redistribution of GM1 and cholesterol lipids. So we believe that these three factors protect the membrane from the action of oligomers. And you can see wild type, these are C. elegans, these are nematodes, so worms. These are wild type worms that move, looking for food all the time. When we have worms, transgenic worms expressing alpha-synuclein, they still move, but very slowly. If you look at the worms, they can still move, but very slowly. When the worms were treated with trodescomine, again expressing alpha-synuclein, but fed with trodescomine, they can move very well. So we had evidence at that time that even in a simplified animal model, trodescomine is protective. And following these results, squalane has entered human clinical trials in 2018 for the treatment of Parkinson's disease by the company entering, founded by Michael Zaslow. Just completed phase 2B that was completed successful. So my colleague, Michael Zaslow, who founded Entering, is now looking for funds and looking for pharmaceutical companies to start a phase 3 trial, which is, of course, the most time-consuming, money-consuming phase and the most difficult to achieve. So he's still looking for funds. That's all. And science is collegial, so I couldn't do anything of these experiments, couldn't obtain any of the results of this presentation without the help of many valued collaborators throughout the world. And thanks to all the people in Florence who collaborated to this project. The group in Genoa, you see, was extremely helpful and deciding this acknowledgement. And all my group in Florence, and that's it. Thank you very much for your attention, and sorry if I was a little bit late.
One quick question, if you have any. Claudio.
Thank you for the nice talk. It's always a pleasure to hear your presentation. I have just a question on polymorphism. That is one fascinating thing about amyloid fibrils. You show a lot of structure that, and some of them has been defined by cryo-EM. Since cryo-EM needs this large averaging on identical structure to reach this high resolution, this structure has been found in different experimental sections, different laboratories. I mean, the best technique, or maybe that we need something more?
No, it's an absolutely important question. So I can tell you that generally, if cryo-EM experimentalists work well, they should try to identify the same species, not just in one fiber in their sample, but in many fibers, and have an overlap and make sure that what they are observing is similar. Occasionally, particularly in patients, when you have the highest variability, they also found 2 or even 3 structures, okay? And so they published that even in the same patient, they extracted different fibers, although there were some similarity in the fibrils. But another colleague for A-beta, he published in the same paper that he analyzed 2 different patients with the same disease, basically it was Alzheimer's disease. So the clinical profiles were indistinguishable, but they found completely different structures. So as a sign of polymorphism. But even in the same patient, even in the same sample, if you induce it, you can have polymorphism. People have published papers where they find even different morphologies, not just different structures. Absolutely. Oh, absolutely. That's why it's important to average many different structures to make sure that what you observe is. But it's a problem. Yes, it's a problem, and I'm sure that this happens in many cases, that people just found, for example, one that was amenable to the analysis. They characterized that fiber, but maybe there are more in the same sample. The dominant species, or the species that was most amenable to analysis, which is even more serious. Yeah.
Fantastic. So let's continue our discussion over snacks here, and let's thank our speaker again.
Quindi quando me l'hai dati ero già 5 minuti dopo. Vabbè, ho capito. Forse non ti guardavo e te provavi. No.